So everybody's still on their graph builder. Should look like this. So in this instance, what I'm going to do is I'm going to take the top one. So I'm going to take my day post-infection, the one that has that blue triangle in front of it. I'm going to take this and I'm going to drag it to my x-axis. Okay. Now what we can do is, obviously we've got all this data together. What we can do is we can look at genes now, right? Anybody have a particular gene they would want to look at according to this data set? Any ideas? What would you want to look at?
The one that's changed the most.
And we are going to find that. But here's what we can do is so let's look at the ACE2 that the virus uses to bind to get into the cell. Let's take a look at that one. So what I'm going to do is under graph builder, you'll see the undo, start over, done. Under my columns, you'll see this search tool. I'm going to put my cursor in there and I'm going to type ACE, ACE2. All right. This gene comes up because it does have ACE2 in it, but we want this one. So what I can now do is I can take this and put it in my y-axis. Okay, this is basically giving you, the default is they give you a curve line to kind of represent the mean across all the time points. These, again, are all your variables. All these points come from your 74 different biosets. Okay. So what we can see is, so 0, anything, again, this goes all the way to negative 100. We can see that after infection, this particular gene goes down. Again, this is the viral way into this cell. You just see this exponentially, like maybe the cell's going, hey, we don't want this virus. Let's down-regulate the way it's getting in. That's probably a good thing to do. What we can also do, I have a buddy who, one of my collaborators just loves straight lines. He gets so mad when I do the curved. I do like the curve, but here's what we can do is click up here and let's hit so on this top menu here, we've got all kinds of options. I want you to click this line. It almost looks like a lightning bolt. It's right next to our bar. Click on that. All right, so now all it's doing is giving you the average. What we can also do now is, so anywhere on the graph, find an empty spot, I want you to right click. Okay. Once you have right clicked, go to add. And we're going to go to add points. Okay, so now we don't have the curve line. Now we're basically just going to the average of all these different points. So wherever that point is the average of all these. These points kind of annoy me. I mean, what I want to do is take them out and just see the average point for each time point. So over here on points, so if we go to the left, where you search for your genes, you'll see this line here, and then down below, you'll see points. And the first option you have is summary statistic, and it's giving you none. It's showing all the points. What I want to do is click on that, and I'm going to select the second one, which is mean. You could do median, you could do whatever you want, but for this instance, I'm going to do my mean. Okay, is everybody with me?
So I don't see the points drop-down menu. I'm not sure why, but it's not showing up for me.
Okay, and you went on to the space, you said add points. Did you put that in?
I did it again and it worked. Thank you.
Okay, great. All right, cool. So we have this, we have our points. Now what we can do is we can put our error bars in. So again, under this menu here where you're seeing points, under error interval, it's doing auto. What I want to do is I want you to select standard error. You can do standard deviation if you want it. I prefer standard error. So click on that. So now here we're seeing all 74 biosets. This is what this ACE2 gene is doing in this particular bioset. And you can see it goes way down as soon as infection happens. There's a lot of variability here, but you can see that once it senses the virus, it's probably going, oh crap, we need to shut the door, right? So it's probably down-regulating this ACE2, and maybe it's coming back up later on. But we can start subdividing this now, right? Go over to your left. I'm going to click on this. I'm going to get rid of this ACE2. I'm going to hit this X bar, or you can just delete it out. I'm going to hit this X. Okay, now our other menu comes up. We are not filtering anymore. Now what we can do is go to your third selection here, age group, and let's just drag it into the overlay. Right? Now what you're seeing, the different colors represent the different age groups in our metadata. And you can see actually it's the old animals that are actually dropping the ACE2 much more than the younger animals do. And you can see at least at this half-day time point, like young animals see no change in this gene. But in this one, you're seeing it go down. So that's what we can do. We can actually use this to investigate individual genes. I could add a gene to that if I wanted to, and we'll do that later on. But again, this is what you're doing. I could take my PFUs. I could see, is there any change in the response based on, different initial concentration of viruses used.
So on the y-axis, the numbers are just relative.
Right.
Normalized relative decrease in expression.
Yes, correct. And again, remember, this is a decrease compared to controls. So it's not just remember, we're not looking at the raw data. What we're looking at are the ranked change in gene expression.
Right.
So if this one was
infected and the infected.
Correct. So what this is showing you is, so normal animals would be at 0. With no change, everything would be 0. After infection, you're seeing that in this particular gene, ACE2, we see a decrease compared to controls in these animals. And the decrease in control seems to be more severe in the aged animals compared to the young, which are in blue, and the old, or the adult animals, which are in red. Okay, so all we did now is we just picked a gene we knew was important. And honestly, in bioinformatics, that's most of my job. And I please don't be these people, but I'll know somebody that does this huge RNA expression, RNA-seq, looking at all genes at the same time. The first question they ask me is, what does their gene do in the experiment, right? You're like, you've got 25,000 genes that change. You want to narrow into something you already are interested in. I get it, but really what you want to do is you want to have the data lead you. Don't lead the data. So in this instance, we led the data. So here's what we're going to do is we're not going to lead the data now. We're going to find our own genes that are cool, okay? So here's what we're going to do is, so all you got to do is get back to the original data set. An easy way to do that if you're on Graph Builder is to go to the top right, and you'll see this kind of jump looking guy. It says show data table. And if you highlight over it, it'll kind of tell you what it does. All I'm going to do is just click on that, and I'm back to my data table. Okay. Now here's what we're going to do is we're not even going to look at the time points now. What I want to do is I just want to find the things that have the highest average change in expression over the entire 7 days and things that had the lowest change in expression over the 7 days. To do that, what we're going to do is we're going to go to tabulate. And actually what we want to do on this top menu here, I want you to click on Analyze. Okay, so everybody clicked on Analyze. You see all these different variables here. What I want you to do under the menu Analyze is click Tabulate, which is the third one down. So everybody hit Tabulate. You should see a window like this come up. Is everybody here? If you're not, let me know. Okay. So now what I want to do is basically over all those data sets, so 74, I want to find the mean of all the different genes, right? So I'm going to take, so in this window here in the very middle, you have different things that you can calculate, different variables that they give you that you can do. You can do min, max, whatever you want. What I want is the mean. I'm going to select it here. I don't double click it. I just select it. And then I grab it. I drag it to the top zone for columns. And you'll see a mean up here. You should see a mean. Now what you're going to do in these columns here, we've got 10,628 columns to choose from. What I want to do is get all my genes. So I'm going to select all the genes. So I'm going to go down here. Here are all my variables. And then when we start getting into these numbers here, these are actually the genes themselves. So I'm going to select this first one. And then I'm going to take my menu. I'm going to take my slider. I'm going to go all the way to the bottom. I'm going to select Shift, and then I'm going to click on the last one. So I'm selecting all of my genes. And remember, I have a magic button. If you get lost, you can always hit the magic button. All right, so I've selected all my genes. What I'm going to do now is I'm going to carefully grab them, and I'm going to put them in my box, right where that mean column is. And it might take a little while. Does everybody's column look like this? Oh,
Mine
go ahead.
looks this ways. So how do I make it this ways? Put it in the
Wait.
one over. Like mine's horizontal. Do I need to undo and put it in the like left-hand mean box?
Right. So let me undo one. So again, we have our mean. Your mean should be in the top box. I've selected all my genes, and what I want to do is don't put it in this top one. What I'm aiming for is this box here. So it's this. So you see 4 boxes. I really want to put it in this like the one on the very bottom left-hand corner. So I'm dragging these, and I'm going to stick. It's hard to see, but you can stick your thing right there. And you should get this. If you can, or you're still having problems, again, you can always go back to your worksheet and click on Tabulate Gene Means. So here's our worksheet. The 1, 2, 3, 4th one down, Tabulate Genes, push that green one. You activate a script, that'll get you to that.
Does the mean reflect that score that it was given by Illumina, or what does the mean reflect?
The mean is the mean of all samples. It's the mean of the Illumina score. You are absolutely correct. So remember, this is our scaled data. So if it's negative, so this gene on average went down with infection. And if you see some of these positive ones, they went up with infection. All right. What we can also do is, I think we're taking the mean of the 0 time point 2. We can go back to our data filter. If you can't find that, again, rows, data filter, and we could say we can only select those time points, everything except 0. So we can go 1. You can see as I select it, the means change as I do that. Select one more. So all we're doing is we're averaging all time points for all biosets for all genes. So this is like, you know, if you're going to rate these genes, this is the one number I want to give it to say, you know, this is what this gene does over this time point. I mean, this is just basically what I'm trying to do is find things that are the highest and the lowest.
I'm sorry, mine won't stop going horizontally. Is there a way to make it go under mean vertically?
I did show you that. If you can't do it yourself, or if for some reason you're having a hard time with that, go back to your original bioset and just hit the green arrow to tabulate gene means.
Okay, thank you.
I don't understand why you're, yeah. If you guys have questions or something, you know, in between now and Thursday, go ahead and email me. You know, you can give me we can do a web conference or something. And I can like work out some of these things with you.
So is there a way we can like order this mean column so that we can like see the biggest and smallest?
Yes, absolutely. That'd be silly if we had it here. So here's what we're going to do is now we're going to make this our own data set. So now that you have this open, we're going to under tabulate, you'll see this red arrow. I'm going to click it. Here's what we can also do is if we wanted to save the script, and this works for even when you're doing graphs, we can save this script. We can say to data table, and that's how I save those green arrows up there, is basically you say save script and you can save it directly to your data table, and you click on that and it will save. As soon as you push that, it'll get you exactly to where you were before. But in this instance, what I want to do is I'm going to click this red one and I want to make into a data table. Everybody see that?
I have a quick question really quick.
Sure.
Do you want us to proceed after filtering out the 0 on the DPI or not?
Yeah, go ahead.
Okay.
You're going to get the same answers no matter. I mean, the numbers are going to be a little different, but the order should be the same. But
Okay.
yes, let's just do. Use the one you did that does not have the 0 in it.
Awesome. Thanks.
Yep. Okay, so I'm going to click on this, make into a data table. So now we have, here's our genes, and here is the mean change in expression of each one of those genes over all 74 biosets. What I'm going to do now is I'm going to select the mean column. And the nice thing about JMP, it kind of works a lot like Excel. Like if you're good at Excel, you'll be very good at using JMP. Trust me. So what I'm going to do is I'm going to select this and I'm going to right click and I'm going to go down to sort, which is the second to last one for your options. And I'm going to hit, I want to sort descending. Did everybody do that? Once we do this, now we have a list. Now we can manipulate this any way we want. The highest gene with the greatest average change in expression compared to controls is the gene MX1. And if you actually look down here and you start looking up these genes, like all these OAS ones, they're all antiviral stuff. You've got chemokines here, cytokines, interferon stimulated genes. Like this makes sense. Interleukin 6 that we mentioned, you know, in the host response, that shows up. But let's focus on MX1. I know a lot of people, and that's why I wanted to do this, is a lot of people use bioinformatics to find biomarkers. Let's see if MX1 would be a good biomarker to maybe as for a reflection of the, you know, how severe is the infection. So let's take a closer look at this MX1. So go back to your data set and go back to our original data set, wherever you put that, SARS-CoV mouse lung. I am going to, as soon as I selected my data set, my data filter is coming up. I want the 0 back in. So I'm going to click 0. We've already created a table for it. Or all you could do too is you could just say, I want to clear all my data filters, and that'll get rid of everything. So we have no data filters on here. Go to up here on our graph. Let's go back to Graph Builder. So again, upper menu, Graph Builder. Under Graph, first option, Graph Builder. Okay. Again, like I said, I like putting the day on the x-axis, so let's do that. So your first variable shown up here, day post-infection with the blue triangle, I'm going to grab that and I'm going to drag it to the x-axis. I also want, instead of columns, instead of these dots, what I want is I want a line. So I'm going to go up here on my top menu options and I'm going to hit line. And remember, it's that kind of the one that looks like a lightning bolt. Click on that. Okay, everybody do. We can also, if you go into your graph, just click on any part that's open, right click, and I'm going to add points. Okay. Now what I want to do is I want to find that gene, MX1. So I'm going to go to my Finder menu up here, click on this, and I'm going to type in MX1. It might take a while. And the faster your computer is, the faster it's able to find this stuff. So hopefully everybody's MX1 has come up. Okay. Once you've seen the gene MX1, I'm going to take this and I'm going to drag it into my Y axis. Okay, I'm going to put that there. I'm going to let it go. So again, we can see we don't get a change at half a day after infection, but after that, again, remember our highest value you can possibly have is 100. And you can see that this gene changes quite a bit. You get kind of peak expression and then it seems at least the rank change goes down. What's really happening, and I've looked at this data set a lot, you get these cell cycle genes start coming up towards the end, like cell cycle gets just ramped up. I think it's repair or something, but a lot of these inflammatory genes actually start getting not as ranked high because these changes in gene expression or this change in cell cycle, like induction of cell cycle just ramps up. So you might actually be, for raw levels, you might be the same level, but at least as its importance as far as like its ranking goes, it actually goes down after this two hour. Okay, now we can see, well, how do our variables affect this gene? We can click on this x-axis, right? Let's pick, now we see all our variables available. Let's pick this third one, age group. I select it, and then I want to drag it over to overlay. Okay, and now we can see what's really interesting here is, so we've got the young is blue, the aged is green. Again, remember, we don't have any time points in the age past 2 because they die, but you can see the response as far as this gene goes and its rankings are almost identical in the young and the old. So maybe this is a good biomarker. It's not going to be affected by other variables. The aged or the adult animal is a little different. We have some different time points for that. What we can also do is, again, we can go to points. We can make this a little prettier. So you've got your line options here. Go down to points. We can select the first one you have is summary statistic. We can click mean. So now we're just showing the mean points. And then the one right below that, error interval, we can select standard error. So this actually might be a pretty good, biomarker. I mean, if you get the disease, like it goes up a ton, right? We can also look at this, and I'm going to kind of skip by this kind of quickly, but we can also say, all right, what does the effect does viral dose have on this gene? So we can actually, what I want to do is I just want to look at the adult animals. So I'm going back to my data filter. We can select let's just select adult. What I want to do is I want to keep all my variables the same, but just change one thing. So I want all adult animals, and I want all adult animals that were treated with the most severe form of the virus. So MA15. So I'm going to hold my control down and I'm going to click on that. And now what we can do, instead of, these are all adult animals, having an age overlay is not it doesn't really inform us at all. What I can do is now I can select viral dose, 1, 2, 3, 4, fifth one down, PFU, viral dose PFU. I can take this and drag it into my overlay. And again, here on your right is giving you the colors of those. The purple is the one with the highest initial virus that we use to infect these animals. You can see the 100,000 and the 10,000 are pretty similar, right? You can see it, and they definitely this gene gets induced in those particular initial things a lot quicker than it does in the lower doses. You can see the red and the blue are actually just almost like identical. They're actually just tracing each other. So you definitely have two different, so what this biomarker is it's actually, not only does it, indicate, the severity of the strain, it gets induced more, but also, the more virus you use, the quicker and the higher that response is. But again, you can see it's like one of the highest changing genes in, all of these. And then if we go to the 7 hour, we can see that, even at the 7 day post-infection, we can see that our purple one is the top one, which is the one we use the most virus to infect them. Green is our second, and then blue and red are all the way down here. So again, this might be a very good biomarker that we can use for our experiment. And if we actually look, so this is what I do is I'll go through, I'll look at these genes, I'll see that tabulate, I will see, wait, where's my table? Nope. Just doing some simple analysis, and there's a lot more stuff we can do. We could sort this descending. I find MX1, I can take that, and then I can look it up. And then I actually looked up that gene. So here, again, this is this is when we graph it. Again, you have the magic note button to do this stuff with. We can see if we subdivide it by increasing severity of virus, we can see that the response gets higher and earlier the more severe the virus is. We can also see that, it's also dependent on the initial concentration or infection of virus. But when you actually look this up, so this MX1, it's this MX dynamin like GTPase1. I've never heard of it before, but it's induced by type 1 and type 2 interferons, and it antagonizes. It's in viral defense mechanism, which makes sense, right? We would expect these genes that are involved in defending the cell against viruses to be way up. What I also find amazing is that you're also seeing a related isoform or gene family member, MX2, also shows up pretty high on our list. I think this was like 24th, right? Oops. So let's get down here. So we can actually kind of look this up and see what this gene does. Again, they're showing it goes up with this. When we actually look, and actually this is kind of how we figure out, hey, we're onto something. People have also seen that this gene, this MX1 antiviral effector, goes up in actual human patients when you look at nasal swabs, right? That's a great sign. So the highest gene we're finding, people are also finding in humans in the current disease is also way up. Also, what they found, and I find this fascinating, is that in isoforms of this MX1, if you have polymorphisms, seem to make people have better or worse outcomes based on the type of polymorphism in this particular gene. So that's gorgeous. What's also nice too, what I'm saying is this could be a very good biomarker to determine if somebody's infected. Not only that, but I recently found this paper. So what they did was they basically took ACE2, what we looked at, and in healthy tissue, what they found was the 50 most correlated genes to ACE2. So if ACE2 was high, these genes were high. If ACE2 was low, these genes were low. And if we look, this last gene here, again, we're getting OAS genes. Remember, we showed those on our list. MX1 shows up, and sure enough, it's correlated to ACE2 levels in even normal skin, normal epithelial tissue. So basically what this is saying is, this might be a very good biomarker. Not only can it maybe help us determine whether, the initial severity of infection, because, we've seen it change with initial dosage and different strains of virus, but also this might be able to tell people that are maybe would be more resistant to this disease or not, because it seems to be correlated to ACE2. And what they found is people with more ACE2, like older individuals, tend to suffer more with this disease. So again, we've went in, we found a bioset, we found a biomarker, we investigated based on different variables. It shows that it's going to be very good at determining those. And then what we find is a very key, anchor that we've seen associated with this disease, it's also correlated to. And what I find is, it's positively correlated here, but as you can tell, when the actual infection goes, ACE2 goes down, but this gene goes up, which I find fascinating.